It's neuron, typical neuron with pericardium, cell body, and with the processes. You know that nervous cells have the processes, yes? What processes may be presented in the neurons? Huh? What processes in the neurons? No, when? Not prepared, didn't prepare, I see. I see, and I understood why you disappeared suddenly. Your name is? Priya. No, what Priya? Sorry, what processes? It may be axon or dendrite. Dendrite, one or more, yeah? In sense organs, sensory or receptive cells are the neurons. Morphological type of these neurons. What morphological type of these neurons? Do you remember the classification of neurons? No. What type of neuron is shown here? Bipolar. Two processes. One of them is dendrite and another one is axon. This receptive cell react on the stimuli, generate nervous impulse, conduct this impulse, and then synapse with next neuron that is this sensory receptive. This neuron is functional. What is the role of this neuron? Do you remember the classification of neurons? Morphological and functional classification. Morphologically, bipolar. Functionally, receptive. This one, bipolar, function. Interneuron, associative neuron. So, receptive neuron, associative neuron. Next associative neuron, and so on. Chain of neurons. So, afferent chain of reflex arch will synapse with neurons localized in the brain. Yeah? So, what processes present in this neuron? Axon, dendrite. What process is axon? What is dendrite? This one. Dendrite or axon? You said dendrite. Nervous impulse is transported in this direction. From dendrite to pericardium, from pericardium to axon. Yes? So it's axon, it's dendrite. Dendrite because this cell accepts information, react on the stimuli with help with dendrite. Nervous endings is terminal portion of dendrite or initial portion of dendrite. Yes? So photoreceptive cells in the eye. It's neuron. Bipolar sensory neuron. We studied nervous receptive cells or afferent neurons, do you remember? In the ganglia, spinal ganglia. But these cells are pseudo-unipolar. And I explained you that sensory neurons are pseudo-unipolar in the ganglia, but there are sensory neurons that are bipolar, not pseudo-unipolar. Good example of bipolar sensory neuron is photoreceptive cells in the retina of the eye. Yes? So these receptors is primary sentient because neuron is the first cell in this chain. If the first cell, sensory cell, is neuron, it's primary sentient. Is that clear? In the secondary sentient organs, what cell is receptive cells? It's specialized epithelial cell. Epithelial cell with different structures on the apical pole. It may be microvilli, cilia. Yeah? These cells react on the stimuli. This cell generates the nervous impulses. this cell conduct nervous impulse from apical pole to the basal pole, where synaptic vesicles with neurotransmitters present, and this cell translate information or transport nervous impulse through this synaptic contact. It's synapse, but synapse not between neurons, synapse between epitheliocyte and neuron. You see that in this case, neuron, the first neuron, is not receptive. To say honestly, this neuron is associative already. Yeah? This neuron doesn't react with the stimuli. Epithelial cell react. And neuron, the first in this chain, nervous cell is the first. It's epithelial, it's nervous cell. But this cell is not receptive, associated. Yeah? Sometimes we name this cell as afferent neuron, but in reality, afferent means sensory. It's afferent. It's interneuron associated. So this principle of organization corresponds to secondary sentient sensor. Why secondary? Because neuron is the second, the first is epithelial cell. Clear? It's the principle of classification of these organs. So to classify, we must reveal what cell is receptive. Neuron, primary, epithelial, secondary section. Clear? It's not so difficult, yeah? Well, now, in what way receptive cells, neuron, epithelial cell, in what way these cells react on the stimuli? In what way these cells may be excited by the stimuli? What is the mechanism of reception? Mechanism of reception. Do you understand my question? Well, who can explain? How the cells react? How the cells became excited? Huh? It's the surface of the cell, dendrite. Dendrite, membrane, covering the dendrite. And stimulus. For example, the light. Photons. Photons of light enter in eyeball and then react with the dendrites of photoreceptive cells. Dendrites of these cells. In what way nervous impulse appears? Excited. To be excited means nervous impulse must be generated. Yeah? And what is this nervous impulse? What is this excitation? Do you remember physiology, physics? Huh? Nervous impulse. What is this nervous impulse? No? Change of charge. Yeah, membrane changes the charge. There are ions, positive, negative charge ions on the surfaces of membrane. And when charge changes, the difference of potentiality appears in membrane. It means the cell is excited. Yeah? So to change the charge, ion channels must transport positive, negative, anions, cations must transport. So channels must begin to work. When channels begin to transport the ions. What is the main component of membrane that react with stimuli? It may be photon, it may be chemical molecules that stimulate the olfactory cells, or taste bud cells. What? to react, any cell, not only sensory cells, any cell, to react, to respond to stimuli. What cellular membrane, what components must be present in cellular membrane? Receptors. Receptors. And what it is? Receptors. Membrane receptors. What it is? No? What is this? Receptors in membrane. What is it, this? What maybe molecules, what substances form receptors? Huh? Who knows? Who remembers? Huh? Cell. Cell form receptor. It's a cell. It's a part of the cell. Membrane, plasmalem, surrounding a cell. So, receptors are the proteins, molecule of proteins, intercalated in the bilayer lipid, bilayer plasmalem. Molecules of protein integrated in plasmalem. So receptors are the molecules of proteins. And for different stimuli, there are different receptors. Now, for example, do you remember we studied the glycogen inclusions in hepatocytes, liver cells, cells of the liver. Hepatocytes accumulate glycogen, or glycogen. How? In what way? When glucose level in plasma increase, hepatocytes obtain this glucose from blood stream and produce glycogen. Glycogen is polymer of glucose. So, hepatocyte must must obtain this glucose from bloodstream. How hepatocyte understand? Oh, glucose level is high. I must evacuate glucose from the blood, from plasma, and accumulate glucose in form of glycogen. How this cell understand this situation? No, there are the cells that have the receptors for glucose. And when concentration of this glucose increase, these cells begin to produce hormones. These cells are situated in pancreas. So, Langerhans islets of pancreas contain the cells that can feel the level of glucose in the blood. And these cells, pancreatic cells, produce hormone, insulin. And this hormone can react with the receptor on the membrane of the hepatocyte. So hepatocyte has receptors for insulin, for this hormone. And when hormone binds with this receptor, the cell begins to transport glucose from bloodstream into cytoplasm. It's reaction, but this reaction is mediated by the presence of receptors. If receptors absent, the cell cannot react. Hormone may be presented, cell present, hormone present, receptors absent. Level of glucose is rest high. Clear? The same in the sense organs. Cells, receptive cells in the sense organ, photoreceptive cells, for example, in the eye. What proteins, what receptors, receptive proteins present in cones and rods? What? How you name these proteins that can react with photons? No. What? Louder, louder. No. You prepared home task. You said that you prepared home task. What proteins, receptive proteins, are characteristic for rods and cones? Students, shame on you, students. Rhodopsin, idopsins, what is this? Rhodopsin, idopsin, what is this? What is this? Huh? Rhodopsin, idopsin, it's proteins presented in the membranes of the photoreceptive cells. In dendrite of photoreceptive cells, rhodopsin or idopsin may be present. For rods, rhodopsin, rods. Idopsin for the cones. But the membrane of these cells, photoreceptive cells, contains the proteins, protein molecules. Rhodopsin and idopsin. And when the light, photons, enter in the eye, these photons influence on the rhodopsin, for example, in the rods. And what reaction begins under influence of the photons? Rhodopsin is cleaved, dissociate into opsin and retinal. So, chemical reaction takes place when photon contact with this protein. Protein molecule begin to cleave. Yeah. So this chemical reaction start the activity of ion channels. Activity of these ion channels depends on the presence of receptor and how we name the stimuli molecules. I forgot in Russian. Wait. Ligand. No, maybe do you know this term? Ligand. Ligand is a substrate, a molecule or photon of light. Ligand is a substance that contact with receptor. Receptor and ligand. When ligand contact with receptor, in the eye it's photons, contact with protein. Protein begin to change. In the eye it's chemical reaction, dissociation of rhodopsin or idopsin. In the, for example, nervous endings that feel pressure, it may be conformation of protein molecule, conformation of molecule, different changes of receptors, but all these changes, conformation, dissociation, of binding, all these changes provoke the transport of ions. So, ion channels became active when receptors react with stimulus. Is it clear? Channels transport ions, charge of membrane changes, potentiality appears, nervous impulse appears, nervous impulse is transported along of the cell and then through synapse to another cell and so on and so on. So, any receptive cells in sense organs must have receptors, specific protein, specific for light, specific for chemical molecules. You eat the sugar, you feel the sweet, yeah? Or you can put in your mouth, in your oral cavity in mouth, you can put mustard. Mustard? Mustard. No, you understand. Mustard. Different feeling. Sweet and sugar and mustard. Yeah. Different. You feel different sauce. Yeah. Why? Because different proteins specific for sweet, specific for salt, and so. Is it clear? Good. So, you understand that there are a group of organs, system of organs, that help us to react on the environment, on the stimuli. You know that all these organs may be classified into two groups. You know the difference, origin of receptive cells, neurons, epithelial cells. And I hope you know for the moment now, you know that any cell, epithelial, neural cell, can react if receptors present. Imagine you travel in highland, travel in highland, sunshine, clean snow, snow is white, brightness, intensive brightness. What changes in your eye can take place when intensive light activate photoreceptive cells? Huh? Huh? I said the light, photons of light, provoke dissociation of receptive protein. Rhodopsin is cleaved. So if you illuminate the light constantly, rhodopsin dissociates, and volume of this protein diminish, and may disappear completely in this cell. And what will be the subsequence? Yeah? What problems? Huh? When receptor disappear, can you accept the light? No. So it's named snow blind. Why? Because intensive light activate photoreceptive cells, protein dissociates and disappear completely. To restore, we must stay in darkness. Synthesis of rhodopsin is realized in darkness. In lightness, rhodopsin dissociates. But now your rhodopsin dissociates. When you will sleep, at night, rhodopsin will restore. Synthesize. So, if receptors disappear, we cannot see. Yeah? Eyes present, neurons present, light present, vision absent. Because receptors are dissociated completely. Is that clear? Well, now, next problem. Imagine. Sorry, I forgot to. Занят, перезвоню. I obtained the photoreceptive cells from my eye and transplanted these cells in the tongue, in the end of the tongue. I performed transplantation. Photoreceptive cells from the eye, I transplanted in my tongue. Then I take the seed, closed my eyes, open my mouth, and I wait if I will see or no. Will I see or no? Why? Photoreceptive cells present? Present. Receptors, proteins, rhodopsin, present? Present. Why is vision absent? Why? Photoreceptive cells present. Receptive proteins present. Vision absent. Why? And who knows? Who can explain this situation? Rack your brains. No. Well, explain me, in what way you understand that it's boy, it's girl, it's computer, it's the screen, it's lamp, switch off the lamps, by the way, please, switch off, switch off, switch off. In what way we understand, huh? I see, I understand, I see, it's my student, I understand, I see. In what way? We understand the vision, huh? Brain. Brain must work, yeah? Brain. So impulses from the receptive cells, photoreceptive cells, and other cells. Impulses must be conducted into the brain. In what layers the afferent impulses to what layers will these impulses will be conducted, will be transported? In cortex. We studied cortex last week. To what neurons, to what layers of cortex afferent impulses are transported from receptive cells? To what layers? No. Last week we studied cerebral cortex, we studied all layers, and I explained you afferent chains. No. To what layers? Granular layers. Granular layers. Granular layers. So from the receptive cells, associative neurons, chain of associative neurons, and then in the cortex. In the cortex there are the neurons that receive this information, afferent impulses. Then analyze this information, and then send the command to motor neurons. Yeah? You see the auto that approach, you jump. Why? You see, you analyze such situation. Yeah? So, to see, we must have not only the photoreceptive cells, but we must have the intermediate structures, conducting structures, the nerves, peripheral nerves, nervus opticus, optical nerve, present from eye. Yeah? And we must have the brain with a specific region when an analysis of information takes place. In what region of the brain, in what region of the cortex, visual information is analyzed? In what part of the brain? Where? I said you last time. Occipital lobes of the hemispheres. Here, it's center of vision. Center of vision here. So, in our organism, there are complex of structures that form together so-called analyzators. Analyzator. What is this? Analyzator. Sense organ, peripheral part, conducting part, nerves, and central part, cortex. This complex is analyzator. Visual analyzator, analyzator of vision, analyzator of hearing. Cortical center for hearing is temporal part of the hemispheres. So in different regions of our cortex there are neurons that accept afferent information. So analyzator. Try to keep in your mind this terminology. Analyzator. Why? It's important for clinic in future because you have the eye, you have the conducting pathway, nerves, and you have the cortex. And problems may be in each part of this analysis. You will study ophthalmology, and you will see the patients with problems of vision. But it may be problem of the eye, it may be problem of the nerves, it may be problem of the brain. Clear? So the reason of absence of the visual absence of the vision, the reason may be localized in different parts of this analysis, in peripheral part, in intermediate part, and in the central part. Is that clear? So, and you will have to analyze situation, symptoms, to make diagnosis, yeah? So, analyzer, only the eye only, the eye, vision absent. Vision means eye, information to the brain, and analyze of this information, and then different part of the reflex action. Is that clear? 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 Good. Well, now let's begin to examine the organ of vision and smell. We have 2 slides only today, and both demonstrate the morphological features of the eyeball, organ of vision. Organ of smell, you can study with help with diagrams, schemes, pictures. We have none of these slides. And if you will receive during exam, if you will receive the question, organ of smell, you will receive the scheme, and using this scheme, you must explain the morphology of this organ. Schemes are scattered already today. Scheme demonstrate the morphology of the olfactory region of the nasal cavity. This one, here. This one, yes. So using this scheme, schemes separate sheets of paper. Schemes. No, there are five complex. So you change or you shoot these schemes. You will shoot. You can shoot, but don't forget to examine these schemes. Slides. It's for the eye. But before slide examination, I want to explain you the anatomy of the eyeball. I know that you didn't study yet anatomy of the sense organs, yeah? Anatomy of the eye. That's why. It's sagittal or sagittal section. Sagittal or sagittal? Sagittal section of the eyeball. Eyeball is spheric, yeah? And here you see the sagittal section of the eyeball. It's anterior part of the eyeball and posterior part of the eyeball. This part of the eyeball contacts this part, contacts with environment, with air. So it's anterior part of the wall, and here lateral and posterior part of the eyeball. Is that clear? Eyeball is localized in orbit, yeah, in the cranium, in the cranium, in the orbit of cranium, yeah. So all this part of the eyeball is situated in the cranium. Only this part is visible because contacts with the air. Clear? The wall of this organ has 3 tunics. What tunics present in the wall of the eye? Outmost tunic, sclera, dense, regular connective tissue. Then, choroid, loose connective tissue rich in blood vessels, and normally, usually, dense, black, brown, colored in black, brown. Why? Because this tunic, connective tissue of this tunic, contains a lot of melanocytes, pigment cells, with melanin granules. That's why this tunic is usually dark, brown, black. And the innermost tunic, retina. So three tunics: sclera, choroid, retina. We have the slide that demonstrates a part of the posterior wall of the eye where all tunics are visible. But in the anterior part of the eyeball, these tunics are modified. Sclera continues into the cornea, well-known cornea. Cornea is derivative of the sclera. Sclera is transformed into cornea. And what is the difference? Dense connective tissue, dense connective tissue. But in the cornea, additional tissues appear. Stratified squamous non-keratinized external surface, and internal simple squamous epithelium. So epithelial tissues appear in cornea. But it's a part of the external tunic. It's a part of outmost tunic. Sclera and cornea both form external envelope of the eyeball. Clear? Choroid. Choroid, in the anterior part of the eyeball, choroid is transformed into ciliary body. Ciliary body is choroid, transformed choroid. Connective tissue, blood vessels, but additionally here muscle cells, smooth muscle tissue appears. In choroid, smooth muscle tissue absent. But in the derivative of choroid, in ciliary body, muscle tissue present, well developed. It's muscle of the ciliary body, involuntary, smooth, can contract and relax. Why? Big question. And then ciliary body continues into iris. 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 Iris is diaphragm. Iris is visible. Iris. 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 Iris is diaphragm. Why diaphragm? Because pupil interior orifice can be enlarged or contracted. Why? As diaphragm in the light microscope. We open, we close diaphragm. The same in the eye. Iris may contract, may relax, and the intensity of light changes. Yeah? So iris contains muscle tissue too. Smooth muscle tissue. Circular bundles and radial bundles of smooth muscle cells present in the iris. Circular, contract, radial, relax the iris. Yeah? And the retina, innermost tunic, covers the inner surface of the eyeball. And what tissue forms retina? What tissue? What? Nervous tissue, yes, neurons, nervous tissue. But besides of nervous tissue, retina contains epithelium. One layer of retina consists of the epithelial tissue, epithelial cells. Retina has epithelial layer and then nervous tissue where neurons are situated. So epithelium is named as pigment epithelium. Pigment epithelium contacts with choroid. So this line corresponds to epithelium. This line. So pigment epithelium of the retina contacts with connective tissue of the choroid here. And the rest part of retina is nervous tissue. Clear? We will see the pictures and slides now. And now look, nervous tissue of the retina suddenly stopped, disappeared. And only epithelium, pigment epithelium of the retina continues to cover the ciliary body, then to cover posterior part of the iris, and then this epithelium contacts with epithelium lining anterior chamber of the eye. It's anterior chamber, posterior chamber. Anterior between cornea and iris, posterior between iris and lens. Posterior chamber, anterior chamber. And here in these chambers there are fluid, liquid, fluid. This fluid, this liquid fluid is blood plasma filtrated from blood vessels in ciliary body. Ciliary body contains the blood vessels because it's a part of choroid, blood vessels present here, and filtration, blood plasma is filtrated and fluid appears in this space. Then through pupil this liquid fills the anterior chamber, and then the liquid must be evacuated through Schlemm canals. Schlemm's canals. It's vein-like vessels presented in the limbus. Limbus is part region between sclera and cornea. Sclera, cornea, this passage is named as limbus, and here in the limbus there are canals, or channels, canals that drain the fluid from the chambers. Is it clear? So, sclera produce cornea, choroid produce ciliary body and iris, retina produce pigment epithelium that covers ciliary body, retina, and contact with another epithelium, simple squamous but devoid of pigment cells, pigment granules. Is that clear? This cavity, is not empty. Here vitreous body present. Vitreous body is jelly-like tissue, transparent. Vitreocytes, cells of vitreous bodies, present here, produce matrix, jelly-like matrix that conduct the light to retina. Lens is suspended by the ligament. Ligament contacts with the ciliary body processes and capsule. Ligament may be stretched, may be relaxed, and concavity or convexity of lens changes too. It's a mechanism of adaptation of the eye. We will discuss later. So it's anatomy of the eyeball. Is that clear? Now pay attention once more. Light enters the eyeball through cornea, then through these chambers, through fluid, through pupil, then cross the lens, and как это преломляется? Dive, no, you understand. And reach the retina. And the light passes through retina, because receptive cells contact with pigment epithelium, not with this surface, but with this one. So we have two slides. One of them demonstrates cornea, well known, you studied already. If you didn't draw the cornea previous time, you can do it today. Cornea. If you draw it already, if you have the picture, no, check once more. Maybe some designations, additional designation must be done. And the second slide is a part of the posterior wall of the eye. So we have specimen of cornea, a specimen of the wall of the eyeball, posterior wall, with 3 typical tunics of this organ. Yes? Now pay attention. It's optical nerve, nervus opticus. You see that this nerve begins from retina, penetrates the wall, and then conduct impulses to the brain. In this region of the wall, photoreceptive cells absent. I show you the slides. Here, photoreceptive cells absent. Only nervous fibers present here. That's why this region is blind. And we name blind spot, or disc of the optical nerve. Disc of the optical nerve, or blind spot. And another special region of the wall. It's axis of the eye, central axis of the eye. And the light that go along of this axis reach the region named as yellow spot. Yellow spot. Yellow spot. Here retina is thin, became thinner. In this region the cones predominate, rods absent, cones are concentrated. Cones contain iodopsins that are yellowish. Molecule of iodopsin yellowish. That's why yellow spot. Concentration of cones. It's the region of the best vision. Vision of the colors. Because iodopsins react with different waves of the spectrum. Short waves, long waves, blue, red, green, yellow, so different length of the light waves. And your iodopsins are specific for blue, for red, for green, and so on. Is that clear? So it's anatomy. And now histology. No, I will not demonstrate you cornea. I hope that you remember the morphology of the cornea. Do you remember? Cornea. Do you remember? No? Do you? Or do you? Do you want, what does that mean in Russian? Do you? Do you in Russian means blow, да? Я дую. Как это? Blue. 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 Blow. Ну, blow. Blow. Я дую. Дую. Ты дуй. Дую. Do you want? Do you? Blow. Do you? Do you know the structure of cornea? Good. Well, let's...